Hello everyone, thanks for waiting and welcome to today's webinar on smart quantification in gene expression, a data-driven journey from result transcription. My name is Judy and I will be your host for this session. We are thrilled to have you join us today. In this webinar, we will explore data-driven strategies to achieve smarter, faster, and more accurate gene expression analysis through optimized workflows. Our speaker today is Dr. Ng, Vazyme Senior Application Expert. We will start with the presentation, followed by a live Q&A session. Feel free to drop your questions in the chat, and we will address them at the end of the talk. Without further ado, let's welcome Dr. Ng to take us through today's topic.
Hello, everybody. My name is Win Seng. You can call me Dr. Ng. And today we'll be doing a presentation on reverse transcription and smart quantification. So without further ado, let's start. So for today's webinar, there will be 5 contents. So we'll start with the basic introduction, and then we'll get to the RT-qPCR workflow and mastering the ΔΔCt method and smart quantification with FMR5S system. So a bit more introduction on our new qPCR machine, as well as a bit of a case study and summary. So first of all, let's get to the introduction part, the understanding gene expression quantification. So why do we quantify gene expression? So first of all, gene expression, it is a result of functional changes, and we quantify it to measure biological responses, such as signaling, and then we can also use it to do disease modeling as well as structural response. So our DNA, everyone's DNA is we're basically the same, right? What makes us different is that everyone's DNA turns on, and the genes in the DNA, it turns on and off at different times, and the amount and the intensity of the genes that's being expressed is different from individual to individual at different moments of their lives. So these are all responses from external stimulus. And what we can do with qPCR here is that we can measure these differences, how much protein is produced. So even if we have the same gene for a particular type of protein, the amount that it is produced, the amount that it is expressed is different from individual to individual. So there are several methods where we can study this, where we can do quantification. The most simple and the earliest version is to do northern blotting. So you can think of northern blotting as as western blotting. Northern blotting is for RNA, whereas western blotting is for protein. So if you're familiar with western blotting, then you know what I'm talking about. So it is separation based on the molecular weight. And then next we have the RNA-seq. RNA-seq is RNA sequencing. It is basically the same as DNA sequencing, except we convert the RNA into cDNA. And lastly, we have qPCR method. So this is the method that we'll be focusing on today. So it is a highly sensitive method, but compared to RNA-seq, it is still not as sensitive. The difference in that RNA-seq, you have to sequence a lot of information. So the cost is very high and the data is very complex. And you have to do a lot of bioinformatics just to understand the results from RNA-seq. But with qPCR, it is instant. You can view the results on the machine itself. And if you want to further analyze it, you can export it into your laptop and do a bit of click here and there. And you will understand all the results very easily because everything is very intuitive. It's all based on graphical representations. And then there's also the difference in terms of the quantification accuracy, okay? So this will be discussed further. So there are 3 types of quantification. So there you have the semi-quantitative, which is with the northern blotting. This one is just visual inspection. You look at the position based on the ladder, and then you have the RNA-seq, which is the absolute quantification. This one you have to do bioinformatics and you have to look at all of the numbers, right? And then with qPCR, we have 2 types. We have relative and absolute quantification. These 2 are based on simple calculations on the CT values. So this one we will talk about later on. Yeah, and in terms of throughput, qPCR is right in the middle. It's suitable for medium throughput. Compared to northern blotting, usually you do 8 or even 12 if you're really good per run. So normally for qPCR you can do up to now you can do maybe 12 samples because you need to do replicates. Sorry, you can do 8 samples maybe because you need to do the replicates, right? So it's considered medium throughput. Next, let's take a look at the difference between absolute and relative quantification. So for absolute quantification, we will take the CT values that are generated from the qPCR run and we'll measure it against a standard curve. So you will need a standard curve for your gene that you are studying. So this one is usually more suitable for R&D work. So when you want to measure the precise viral load, gene copy numbers, and the changes in copy number, so you will do absolute quantification. But if you are doing a routine quantification, routine study, so like let's say you want to test whether the sample has COVID-19 or if it has or if the pig sample is infected with African swine fever. So that one usually you will do relative quantification because with relative quantification, you don't need a standard curve. You are just measuring between you are measuring with the own existing data. So you're measuring from the reference data and the target gene data. Next, let's go to the workflow. So the workflow for qPCR is very, very simple. You will start with RNA extraction, where you isolate the RNA from the samples, and then you need to do reverse transcription and turn the RNA into cDNA. So cDNA here stands for complementary DNA. So why do we need to do the reverse transcription is because RNA is very fragile. So it is easily degraded. So when you do a reverse transcription into cDNA, it is much more stable. And another thing is that when we are doing real-time PCR, we are still doing PCRs, right? So we are doing polymerization, polymerase, right? So for DNA polymerase, it only works with double-stranded DNA. So it doesn't work with RNA anyway, which is why we need to do the reverse transcription step. And then finally we have the real-time PCR, which is the qPCR amplification. So before we do our qPCR, the first thing we need to do is after we extract our RNA, we need to check, is it good or not? So we need to look at the RNA purity and the integrity. So we usually for this, it's very important because for a good qPCR result, you will need good RNA because as the saying goes, rubbish in, rubbish out. So if you have terrible RNA, then you will get terrible qPCR results because it directly influences the efficiency of the reverse transcription as well as the amplification. And you can see from this figure here, so the higher the quality of your RNA, the better the amplification curve. And as well, it's the same as well as with the RNA purity and the integrity. So with better quality, you always get a better amplification. So how do we check our quality of our RNA, right? So usually we'll use a machine called the spectrophotometer. And a very common brand that I'm sure everybody knows is NanoDrop, right? But you might have used NanoDrop, but you might not understand what exactly you are looking at, right? So for NanoDrop, you will get 3 readings essentially. One is the OD260, which is the absorbance of nucleic acids. Then you have the OD280, which is the absorbance for protein, and then OD230 for other impurities. There are usually 2 values that we take note of. The first is the 260 over 280 nanometers. So this will tell us if the reading is between 1.8 to 2.0 for RNA, this is the ideal reading. So for RNA, 1.8 to 2.0, and the changes, whether it's elevated or reduced, that usually suggests a presence of proteins or phenol or some if it is elevated, that means the RNA is of very low concentration. If it is reduced, then the proteins, there might be protein and phenol inside your samples. For the 260, 230, if it is elevated, so it's very high, that means your RNA is very diluted. If it's very low, that means it's contaminated with carbohydrates or EDTA. Working with plant samples, then the sugar will be a problem here, and you can see it here. Because some plant samples have a lot of sugar in it, so polysaccharides in it, and carbohydrates. The next way to evaluate the integrity of your RNA is through electrophoresis. So I'm sure if you work with DNA, then you will definitely know what is electrophoresis. But one thing that we not everyone might know is how do we view the electrophoresis results, right? So for RNA, when you run RNA, you will get three distinct bands. One is the first one is the 28S, the second is the 18S, and the third one is the 5S. So for the 18S, which is the middle one, usually it is thicker and with higher intensity, because in the cell, in the nucleus, the rRNA, 18S RNA would be the highest concentration. So therefore, it will be the brightest in your gel. So for intact, you will see three distinct bands. If your RNA is degraded, just the same as DNA, it will look very, it will be smeared. Okay, so if there is DNA residue, so like genomic DNA residue, then you will see a very thick band at the top part. So this is very heavy, right? gDNA. So it will be very top at the top part here. And if there are protein residues, usually the proteins will be stuck inside the well itself. And of course, you will see a little bit of smearing here and there, but the smearing is quite common unless it's very serious like this. If it's a light smear, it is acceptable because, as we mentioned just now, RNA is very fragile and prone to degradation. So light smearing is acceptable, except as long as it's not fully smeared like this one. So next is the importance of removal, DNA removal. So just now we mentioned sometimes you have DNA residue, right? So we need to make sure that the gDNA is removed. So when you are doing extraction, sometimes you will get the kit that extracts both gDNA and RNA, but most of the time you will use a kit that extracts just RNA. So if you're extracting just RNA, that means that there should be DNAs inside your RNA extraction kit. So if your DNA is not functioning very well, then it is okay, because in Vazyme, our qPCR reagent contains gDNA wiper. So this is like a backup plan to make sure all gDNA is removed. So first, you can take a look at number 1 here. So if you do not have a gDNA wiper and you include reverse transcription, you will still see an amplification curve. Why? Because the qPCR reagent, it works with double-stranded DNA. So if your gDNA is present, then it can still do the amplification, even though there's no RNA in it, right? Because there's no RNA in it, so the result that you get is not accurate because it's not from the RNA, it's from the gDNA itself. So you might still get the amplification curve, but that is not the correct result. So why wouldn't the results be better if we have a smaller CT values without using gDNA wiper? So what this mentioned here is that when you have gDNA in your qPCR protocol, what happens is that the CT value, which is the value that is detected, will be smaller. And you can tell this very easily if you have two bands, as you have two amplification curves, one have a smaller CT value, one with a bigger CT value. That means the smaller CT value is most likely as a result of gDNA contamination. So that is not the result that you actually want. That's not what you want to study. You want to study the RNA. So if you have both gDNA and the reverse transcription, so gDNA wiper is present, so you remove your gDNA and you do reverse transcription on your RNA, then you will get a higher CT value, but that's the correct amplification curve that you actually want, because that's your target gene that you're actually studying from the RNA. So since qPCR is designed to amplify specific target sequences, any leftover gDNA can be co-amplified along with your cDNA. And this leads to a false positive and inaccurate quantification. So it is very much necessary to remove the gDNA. Next is when we are doing the reverse transcription, we also will need something else, which are the primers, right? So there are three main types of primer. So the first one is the oligo-DT primer. So these primers, they bind onto the poly A tail. So in your RNA, you can imagine this as an RNA. At the end, there will be a poly A tail, which is a lot of A's, A. So with the oligo-DT primers, it binds specifically to the A tail. Why? Because T binds to A. If you remember your ATCG, so T binds to A. So this is where it binds. So the binding site is at the 3' end of the eukaryotic mRNA, and it can synthesize full-length cDNA. So when the RNA is converted into the cDNA, with the oligo-DT primer, it can synthesize the whole length of the RNA, which it does a full length of cDNA. However, it can only amplify mRNA with poly A tails. So for example, if you have fragmented RNAs, so your RNA is not high quality, it's fragmented, so maybe it's cut in the middle, so it doesn't have the poly A tail, then the oligo-DT primers will not be able to sequence any fragmented RNA. So it can only sequence full-length RNA. So that's where the other types of RNA comes in. So the first one is the random primers. So for random primers, it can bind onto any random site along the RNA. So it can synthesize highly fragmented RNAs. But the problem is that it has a poor specificity and you will produce a lot of tiny fragments. So it might not be ideal. And then next you have the gene-specific primers. So this one, instead of randomly binding on random sites of the RNA, it only binds to specific sites. So with gene-specific primers, you need to design it based on your specific target region. So that's the additional cost there. And you can only synthesize specific sequences. So you get less results then, basically. So when we are doing qPCR, usually we won't just use one primer. Usually we'll use two. We'll use the oligo-DT and the random primers. So what happens here is that we can get both the full-length cDNA as well as the fragmented cDNAs. So you will get like a combination of the longer and the shorter ones. So with random primers, you ensure to generate full-length cDNA and avoid missing important RNA species. Okay. Next is the amplification curve. So this is the most important result that you will look at when you are doing qPCR. So first, we need to understand what we are looking at first, all right? So on the y-axis, you will see the fluorescence. So this is the intensity of the signal. So it represents the real-time fluorescence intensity during the reaction process. So as the amplification is happening, each point that is measured in real time. Okay, so you can measure at which exact point the amplification picks up. So the higher it goes, the higher the intensity, the more amplification is happening. That's where it's picking up. And the x-axis represents the number of the amplification cycles. So what the amplification cycle here mean is that the higher the amplification cycles, the longer it has been, basically. So you can measure it as like time in a sense. Okay. So like, for example, if you remember back when we had the COVID, right? So when your CT value is lower at the specific value, that's when people will tell you, okay, you have COVID. If your CT value is very high, that means you don't have COVID, because it takes a very long time for the amplification to be detected. All right, so the lower CT value, the more richer, the more gene is present, the more RNA is present, so it's easier to be amplified. That's how you can understand the CT value. Okay, so for the shape, usually we want a nice curve shape, and like an S curve, because if it is a lot of jagged lines, that means there's a lot of noise. So, and then next is amplification efficiency. So the slope, so this is the slope, right? So it tells us how efficient the PCR reaction is. So it should be close to 45 degrees here. And then next is the initial temporal amount. So if the point where the curve starts to rise, it indicates the initial amount of target DNA inside the sample. So when you in your sample, you have a lot of different kinds of DNA, but what is talking about here is the target DNA, so your target gene that you are looking at. So the more gene you have, usually it also means the lower the CT value will be. And next is specificity and background noise. So the curve should be smooth and free of unexpected peaks of fluctuation. So like what I mentioned just now, it needs to be a smooth F shape. So any irregularity suggests amplification or background noise. So this can be like maybe your master mix is not exactly pure, or maybe it can even be your equipment itself might have some issue. So that would require additional troubleshooting. Next is reproducibility. So usually when we do qPCR, we'll do replicates. We'll do 3 replicates to make sure that the results are consistent. Okay, next is we'll talk a little bit more detail about the CT value. So like as I mentioned just now, the CT value, it represents the cycle number at which the amplification curve reaches a specific fluorescent intensity. So this is the fluorescent intensity. Let's say the CT10 means at this point, okay, we know that it had reached this intensity. So the signal is high enough at this CT value. So why is the CT value important? Because it is inversely related to the initial amount of target DNA in the sample. Like I mentioned just now, the higher the amount of your target inside your total RNA or your total DNA, then the lower the CT value is going to be. So a lower CT value indicates higher initial template. And usually, if the range is within 15 to 35, then we will accept it as a credible CT value. So this is usually where the CT value will be. But how do we interpret the CT value? So this one is difficult because I cannot tell you which CT value is positive, which CT value is negative, because the expression level of each gene, of each sample might be different. Okay, so that is why we need to do controls and we need to do replicates, because each run will produce a slightly different result. It will produce an expected result, but the exact CT value will be different from run to run, from sample to sample. So we compare the CT values of different samples to determine their relative gene expression level. So that's why we have to compare between different sample and between the replicates itself. So the more consistent the CT values are, the more reliable our results will be. So some of the factors that can affect your CT value are primer design, so whether it's a good primer or not, whether it can bind to your target gene or not. And then you also have the reaction conditions, so like the temperature and all that, and as well as your sample quality, like I mentioned just now initially, if your RNA is terrible, then your or your concentration is very low, then the sample quality is bad, right? So your CT value will be very high. So next is the ΔΔCt. So what is ΔΔCt? We'll talk about this now. So from CT to fold change, we can understand based on the study of the ΔΔCt value. So first of all, we will look at the first value, which is a ΔCt. Delta means change. So the change in the cycle thresholds, change in the CT value there. And then next you have the ΔΔCt with the change in the change of the CT value. So this will measure something else. And then that's the third value. I'm not going to pronounce it, okay? So first, let's take a look at the meaning of CT value. So the lower the CT value, the higher the expression level. The more intense, the more gene is, the more DNA is present for the target gene. However, CT values cannot be directly compared across samples due to variation in RNA input and reaction conditions. So like I mentioned just now, it varies from sample to sample, right? So how can we use this value then? So first step is to normalize the delta CT value within a sample. So that's where we do the ΔΔCt calculation. So you don't have to remember, you don't have to do this calculation yourself. All these calculations are done automatically in the qPCR machine. So all the modern qPCR machines can do this automatically. It doesn't matter which brand you buy. Okay. So just a brief introduction here is that ΔΔCt is you take the target gene CT value minus the reference gene CT value. Okay. The next one is you compare between the samples from sample A and sample B. That's where you take the CT value of the sample and minus the CT value of the control, the change in the CT value. So for the control, it will be minus the control gene. And then for the fold change calculation, this one, again, you don't have to do it yourself. This will be done on the machine itself. So if the value is more than 1, then it's an up regulation. If the value is down, then it's down regulation. So what that means is the up regulation is expressed more, down regulation is expressed less. So next is selecting a suitable reference gene. So one of the key components in your qPCR master mix is your reference gene. So usually when we choose a reference gene, you want it to fulfill a few conditions. The first is it needs to have a large amount of expression. So no matter which sample it is, the expression value should be high. It should be stable. Okay, stable expression in different types of cell and tissue. So because you want the value to be more or less constant, the CT value, I mean. And then next, the expression level is independent of the cell cycle or whether the cells are activated or not. So when you extract the cell and extract the RNA, the cell itself might be at a different cycle, different life cycle of its stage. So the expression level needs to be same no matter which cell, which phase of the cell is, if for a suitable reference gene. And then next is that it should not be affected by any endogenous or exogenous factors. So whether for example, if you collect the cell sample from a pig, so whether the pig is sick or healthy, whether it has eaten enough food or not, whether the pig is stressed or not, the amount of RNA produced for your reference gene should be the same. So it's not affected by any external or internal conditions on your sample. So and then next is there are no pseudogenes to avoid contamination of genomic DNA amplification. So pseudogene is it is usually produced as an RNA, but it's not an actual gene. It does something else. Okay, that I will not talk too deeply into this on this webinar. So in general, it is customary to select some common housekeeping genes. So these are the genes that is being verified across different sample types. So the first is the beta-actin, and they have actin-2, GADPH, and ATN's rRNA. So these all serves as a standard internal reference gene. Next is to understanding the ΔΔCt. So this calculation, as I mentioned just now, you don't have to do it yourself. This is fully done by the qPCR software. And at the end of the calculation, you will look at this. So that if it's 1, that means it's neutral. So there's 1 for the target. So 1 is, you don't have to look at this because it's for the control. For treatment, so that is your target, that is your sample, right? So 6.5. 6.5 means that it is is expressed more because it's more than 1. Next is some of the common pitfalls in data interpretation. Some of the common errors that you will face is the variable under treatment, variation under the treatments. So your ΔCt variance is less than 0.5 and the curves are not parallel. So the curves are not parallel, meaning that how to say, the shape is not the same basically. So it needs to be it needs to have, how to say, equal distance. Next is let's take a look. Next we will look at the reference gene instability. So the reference gene expression is affected by treatment. So from treatment to treatment, the reference gene expression will be different. So this will have cause inaccurate normalization. So an example is that the GADPH, which is your reference gene, may be upregulated under metabolic stress. So metabolic stress means like it is sick, and then it causes an apparent downregulation to target gene. So if it's sick and then it doesn't produce as much. So this is what we mean by instability. So you need to validate the stability of the reference gene. So you need to choose the correct reference gene. Next is the high variance in replicates. So from replicate to replicate, there's a lot of differences. So these differences might be due to pipetting errors or sample inconsistency. So pipetting errors, okay, you know what that means. That means when you're pipetting the master mix, you're not pipetting correctly. Some of the reagents are stuck in the pipette tips. And then sample inconsistency could be due to, example, when you're doing RNA extraction, you do RNA extraction for sample 1, sample 2, but then the amount of input you put in is different. So you get a sample inconsistency or the degradation. So one of your sample is highly degraded compared to the other. So what can you do is you have to check your pipetting accuracy, sample homogeneity, so like the concentration and the ring value should be the same, and the PCR efficiency. So PCR efficiency here might be less. If you're using a master mix, then usually you don't have to worry too much about PCR efficiency because the master mix is pre-made, right? Unless you are mixing your master mix yourself, then you will have problem here. Next is nonlinear amplification. So this is the parallel thing that we are talking about just now. So the amplification curve shows tailing or non-parallel trends. So it's not parallel, then it indicates reduced efficiency or distorted CT values. So the solution is to maintain the amplification efficiency between 90 to 110% to avoid reaction inhibition and tablet overload. So what this means here is that you want to make sure you have enough master mix, basically. You want to have enough DNTPs, enough primers. You want to add more than you actually need. Because I know some labs you want to save on cost, so you reduce the amount of master mix. But at some point you reduce too much, then it will not be able to maintain the amplification efficiency. Next is smart quantification. So this is where I will introduce our new FMR5S, our new qPCR machine. Okay, so some of the features here include is the 5-second full plate detection. So the detection speed is very high. And we also have preset program. So it's very intuitive, very easy to use. Just click on your selected program, click run, that's it. Next is the consistent built-in ΔΔCt calculation. So this is where I mentioned just now, all of the calculation will be done automatically in the machine itself. You don't have to calculate anything yourself. You just have to look at the results. And one good news is that this software, it is installed onto the machine, but you can also install it onto your own personal laptop, so which we will provide when you purchase the qPCR machine. And you can analyze it afterwards on your own time. And next is experimental setup. So this is just a brief introduction on how we usually set up our experiment. So I mentioned just now, we will always do replicates, right? So because you need to check between, you need to check for consistency. So there are a few things that we will look at here. The first thing is NTC. So NTC stands for no template control. So this is where you add your primers and your master mix, but you don't add your cDNA template. Okay, and the next one is NRT. NRT is no reverse transcription. So this is where you don't add the reverse transcription mix into it. So you only add your RNA template. So this is where you want to check for gDNA contamination. Because if you don't add the reverse transcription mix into your sample, then if there is amplification curve, so because the amplification will not exist if it's from RNA, so it can only exist from the double-stranded DNA. So if it exists without the reverse transcription, that means you have gDNA contamination. Okay. Next is you want to choose your technical route. So you want to choose whether you want to do absolute or you want to do relative quantification. And then next is the technical replicate. So this one is where you do 3 replicates. And then biological replicates, so you will do two, you do more than one groups basically, and then experimental replicates, so overall reproducibility. So the setup usually looks like this. You will have your target genes and you have your reference genes. Okay. And then next you have group 1, group 2. So these will all be replicates. So you don't have to think about this too much. You just have to do it like this. And then group 1 is sample 1, group 2 is sample 2, basically. And then you will have your NTC and you have your NRT. So these are your NTC is your no temper control, NRT is your no reverse transcription control. So this is how you will set it up. So basically it will be group 1 would be your 1 sample, basically. Next is the plate setting. So this is you can do this directly natively on the software itself. So the plate setting, you can do it while the machine is running. Before the machine runs, before you start it, you can set it up, or you can also set it up afterwards if you remember where you place all of your samples. So this one can run 96 wells. 96 well doesn't mean 96 samples because you have to do all the replicates. And then you can also do your result analysis, so they can be done natively on the software. You can do it on the machine or on your own laptop. Next is the summary. So for Vazyme, we provide a full solution. So we start from the nucleic acid extraction, assay setup. So we include the machine as well as the reagents itself. So this we have R433, which is the all-in-one super mix for qPCR. So this is a reverse transcription kit, and then you have the real-time PCR. So this kit is for the real-time PCR. So these are separate steps. And then for the assay setup, we have you will run it on our new qPCR machine and the result analysis on the software. So a bit of an introduction on our products. So because we are doing automation, right? So we will introduce you on the automated RNA extraction. So we have 2 kits. One is the RMA101. So this is suitable for animal tissue. And then you have RMA4101. So this one is for plant extraction. So this has a broad compatibility. We have a very comprehensive list of samples that we have previously tested and verified. And then for the machine itself, we have 2 machines. We have VNP96 and 32. So they both do the same thing. They are automated extractors. One is a 96 sample, one is 32 sample. So if your lab has a smaller footprint, so we will recommend VNP32. If you have a very large throughput, then you can go for the 96. So these are all automated, and it comes in these cartridges. So these are for the pre-treatment. And once you have pre-lysis, pre-treatment it, you load it onto the cartridge and load the cartridge into a machine, and it will run smoothly. And you just need to extract the DNA, remove the DNA at the end, or the RNA here in this case. Okay, so we also have manual options. So manual options would be column-based extraction kits. For plant tissues is RC411. For animal tissues is RC112. And we have a broad list, a very comprehensive list of compatible samples for both plant and animal. And the steps are very simple. For RC112, it's only 6 minutes. So if your lab has a very small throughput, you don't have a lot of samples, then you can go for this, and it can be done very, very quickly. And as for the plant isolation, RNA isolation kit, so it works for both plant and fungal samples. Next, we'll move on to reverse transcription. So this is the step where we turn the RNA into your cDNA. So for this, we have R433. And R433, it is good. It had one of the features that is high reverse transcription efficiency. So we can basically allow you to achieve a smaller CT value. And then the reaction is very, very fast. It takes only 5 minutes from after you load your RNA and your reverse transcription master mix, and you put into the machine. It takes only 5 minutes for the reverse transcription. It's very, very fast. And here we have a validation as well. So because some of you might not believe 5 minutes is enough for reverse transcription if you come from a more traditional setup lab where the reverse transcription takes 10 to 15 minutes, then you, oh, how come 5 minutes works? So we have validation for that. Next is the real-time PCR itself. So this is where the quantification step starts. So for this, we have Q713. And here one of the things that we want to highlight is the anti-contamination system, which is the DUTP heat level UDG system. So what this does is it makes sure that your qPCR reaction will not be contaminated by qPCR reaction of the previous step. So sometimes it's very rare, but sometimes what can happen is that you have RNA, or no, sorry, you have cDNA from a previous run, and then it somehow gets contaminated into your current system, your current protocol, and then you will get what happens is you get more than one amplification curve that you are targeting. So that's contamination. So this DUTP system, it prevents this from happening. But there's a caveat, which is that you need to make sure that the machine only uses master mixes with this system. So you cannot mix and match. You cannot use different PCR mix that does not have this exact system. Otherwise, it defeats the purpose because of the way it works. Okay. Next is that low expression, high precision. So we have a reliable detection and relative low abundance for relative low abundance gene. So if your gene has low expression, it can still be detected with our Q713. You don't have to worry too much about that. And next we have amplification of moderately degraded samples. So sometimes it is inevitable. So your RNA samples cannot always be very high quality, right? So we have a validation here that proves that we can work with even slightly degraded samples. Of course, if it is highly degraded, then it will not work. But if it's just slightly degraded, then it should work. And next is our latest product, which is our FMR5S qPCR machine. So it features 5S, right? What is the 5S? Screen. So you have a 12.1-inch HD touchscreen. Very good to easy to use because it is situated right in front of the machine and you can lift it up. So even why we design it this way is because you want your qPCR machine to have situated at a lower bench, so you can load it on top and you can lift the screen up. So it's very intuitive. Next is smart. It has a 12-column temperature gradient and it can run optimization and happens in a single run. So you can run it. You can do gradient, basically gradient testing with different temperature, and then speed. So the heating speed is very high, so very fast. So it can ramp up 8 degrees per second, and you can get a 5-second full plate detection. So very high speed detection. Speed stability. So this is based on our sensor is based on a LED-based optics. So what LED is good at is because it has a relatively zero maintenance, very long lifespan. So you can use it for a very long time. Your LED will probably still be functioning after your machine is malfunction as a whole. So you don't have to worry about the sensor. Next is the sensitivity. So it can detect very low background, ultra low background signals. Sorry, it can detect strong signals with ultra low background noise. So you can get confident results even with low abundance target genes. So some of our competitor qPCR machine might have issues with background noise. So this is not a problem that we have. So here is a summary of for troubleshooting. So I won't go through all of this because it's going to take too long. So you can either screenshot this or you can just ask us later. I will send you a PDF copy of this troubleshooting. So this is for the abnormal curves and this is for the CT value. So this is very useful when you're doing troubleshooting because no matter which lab you're in, you are prone to face some troubleshooting issues. So this is a very good guide. So a little bit introduction on our company if you are not familiar. So we are our core technology is based on these 7 technologies, which is biochemistry, molecular biology, immunology, enzymology, chemistry, molecular engineering, and bioinformatics. Okay, and for our production line includes life science, in vitro diagnostics, and biomedicine. And we have a very broad range of customers, from scientific research institutes to vaccine manufacturers, as well as medical examination institutions, as well as independent labs. So we cater to a very broad range of customers. And we have a very large product line. So if you have any product that you're interested in, and you might find it difficult, if you find it difficult to locate the exact product, you can always talk to our salesperson, because we have a very broad list. I'm sure at some point, somehow we'll be able to provide the products that you want. So this is the most exciting part of today's webinar. Okay, so as a launching offer, so promotion, we have a 30-day free trial, and you can receive up to 40% discounts upon completing the registration form. So you can get access to the registration form by scanning this QR code, or I will also give you this link shortly in the chat, and you can go to the link and fill in the form. So the form, when you scan the QR code or you click the link, you will see this page, and all you have to do is just fill in these details. Okay, and that's all for today's presentation. I will put this in the